species of larva you are working with, and that will help you decide what products you may need to apply, how soon you need to come back. So it may be very, very familiar with some of you, but we will go ahead and get going. So some of the topics we'll cover is just basic larva inspection, survey tools and equipment, and some of the immature kind of surveillance strategies and techniques, just a couple of tips. There is definitely a knack to it. And it's not, just go out there and take a dip. And then some very basic larva ID, like I mentioned. So some of the equipment that we use, I'm sure you're familiar and have a dipper in. It's basically a cup with a stick on it. Useful for all sorts of things like fishing out golf balls, which I have a much larger golf ball collection than I need since I don't golf. But move snakes out of the road. But we also use it a lot for collecting and looking for mosquito larvae. Another useful tool is larva nets. It's something I didn't use for a long time, and then I really became a fan of. This net here, which used to be sold by BioQuip, you could had a flexible ring, and you could push it through like grates and things if you couldn't access. Unfortunately, we don't have those anymore since BioQuip's not around. So I've been using these little minnow nets. They have little telescopic ends. They're pretty small, but you can usually fit them through a lot of grates. But often what I just use them for is I can pour off some larvae. If I have a bunch and I'm trying to collect some for rearing or something, I'll pour that off. And same as if I'm using the bilge pump, which the cool thing about the bilge pump is you can fit that through grates. If you have a dry well that's 20, 40 feet deep, some papers have put floats on the end if you want to pull from the top, or you can just drain the whole thing and then you eliminate a source reduction. But what I'll do sometimes is pump that into the net over the bucket. So if you have a 5-gallon bucket and you're pumping out a bunch and you don't want to lose all your larvae, you can just kind of pump into the net and kind of condense them. I've never had one of these, but they look really cool. Clark sells a kit, but one of the things, they have these little strainers that you can kind of sort the different instars and things. But one thing I have used a lot, you can buy separate, is there's a little clean water bottle. If you ever remember it, I usually don't. So oftentimes what I'll do is I'll take like a water bottle you get from QT and poke a hole in the top. But some clean water can help you in your dipper cup or something. If the water's really cloudy and you're trying to field ID them, you can just kind of rinse off and clarify the water a little bit. So that can be helpful. So some sites that, good examples of breeding sites. This is one that it's not actually a rain collection barrel. It was somebody's washing machine. They didn't want to run a drain, so it goes into this. And it was a good example of a breeding site because it actually has water that collects or overflows from the top. I have found Aedes aegypti in situations like this. There's a little tiny hole that mosquitoes could actually access the water inside the barrel, and I have seen Culex harboring. I couldn't actually access it, and the homeowner didn't want me to tear his barrel apart because he was just a renter. But I did see that there were, Culex right on the edge of here, so there's a good chance there were Culex in there. And then it leaks from the bottom drain here. It was cracked, and so there was more breeding in there could be breeding here too. So this is like, the worst of all worlds. If you want to use rain harvesting or something, you definitely want to prevent mosquitoes from accessing it, or you could treat it with something like a methoprene or something, residual product, but the best way is to just keep the mosquitoes out. This may look like a more typical site in the middle picture here. You have a lot of duckweed. Sometimes the duckweed will be so abundant that it actually will prevent, they have to be able to access the surface of the water. And so you may not be able to find larvae there because it's just so overgrown. But if there's ever like wind or something creates like a pocket that they can, I have found breeding in spots like that, like in the middle here where there's a little bit of of water that can be visible from the air. And then someplace like a lot of people will, you'll have complaints, may call, be like, hey, there's this giant pond and tons of mosquitoes coming from there. That's probably not true, but water in a desert is still a potential source of breeding. I just wouldn't necessarily think, especially in the middle of a large body of water, especially one with fish. There isn't here, but I also, when I inspected this, you can find these little kind of eroded spots. These look kind of like dead fish. They're actually leaves. But if this, which is right next to this large pond, overflows or percolates through, and there's no fish in this little spot, this could be a potential breeding site. So the complaint probably wasn't correct, but that's still water and it's still worth checking. This could also be overflowing, which it was, into a wash, and the wash was potentially had a breeding in it. So the pond itself may not be worth walking around, but if it has some erosion or something, then or pockets. Also, this was great because I stepped in one, so that's always fun. And then these are kind of more man-made sites that could potentially be breeding, and these have many times. This is where those nets or pumps are useful, because these grates are bolted down and you can't actually access the water. So you'll often find cycles when they turn water on and off. When they turn the water off, these will always have a little bit of residual water in them, and that they will potentially breed. If you can't access it, sometimes you can use like a flashlight to just see the larva, especially the higher instars. This is another similar one, but it releases water into a wash, and then this is what it looked like from the inside. I found a thorax breeding in here, which was very surprising. I would have thought it was just quinks, but I have found quinks here also, and there's a whole bunch of these at this site. And then this is a more typical looking mosquito site, cattails and kind of swampy areas. You'll see lots of mosquitoes breeding here, and I found tarsalis here. And then this is actually my backyard. I didn't do this by accident. I had a toad out and it rained and I saw there was water and I was like, ooh, let's see what happens. So I kept checking it, and this was breeding Aedes aegypti, which I have a lot of at my house. So rain can fill one of these up. Also, the top of this tote next to it had some water on it, and it could be breeding. So this is definitely that container sort of site, tip and pour sort of situation. And then this is a little video. Hopefully it comes through. But basically kind of show like the technique of coming up to a site. You want to pay attention to where your shadow is being cast. Mosquitoes can swim, and they will have a kind of predator avoidance behavior. And so you just kind of want to come in from the side. If you put your, cast a shadow over there, they'll dive and you may miss them. And I've taken new trainees out to areas where I can find larvae, and then they'll dip in the same spot. It's sort of like fishing. They don't find anything. And it's probably because they either were too slow. And so what I always try to do is kind of come from the side, put my dipper from an angle, and then just kind of flip it, turn it so that they'll dive to the bottom of the dipper. And here's another kind of site. So we'll always tell people, larvae can't breed in flowing water. And this, it's hard to see from the picture, but this was flowing slowly. But it also shows there's that exception of if the water slows down enough because of grass or weeds or something, then there could be breeding. And I did find breeding in this spot here. So they were able to hang on enough towards the edges and towards the grass, kind of sheltered them enough that they were able to make it. So I walked or drove this whole wash, and there were a bunch of spots that it was flowing too fast. But then there were spots that slowed down like this and had breeding in it. So I didn't want to pick on Arizona too much. So here's a site that I used to visit on the picture on the right. It was in Colorado, and it was a good example of almost every type of breeding site. So you have a strong, regular flowing river. Colorado River's still have water in them, some of them. And then it has this irrigation channel here, and then it has a big pond with cattails on two of the edges. Then it has this big swampy thing that there's not a lot of surface water with cattails and reeds. And then it has a stormwater feature here with a bunch of rocks, big rocks that would hopefully prevent erosion, but there would often be water in between them with some weeds. And then there's a cemetery. So I would inspect this site, and it's very big. So you'd be thinking, oh, this could take me all day. Where do I take my dips? And it depends. The river, most of it is flowing strong. I wouldn't bother unless it turns and you have water that slows down or overflows into something where it can become stagnant. I would not walk the whole river. But this irrigation channel, it was intermittent. So sometimes it would have water in it, and then it would they would shut the water off, and then the water would be stagnant. So this could be a potential breeding site. Also, when they wouldn't have water, they don't irrigate all the time. It's a good site for floodwaters if it would rain. Sometimes you get water here. And then this site here, which was all cattails, I found breeding here more often than the site over here with a big body of water, because the fish here could still access a lot of these cattails and reeds, and I wouldn't find breeding here as much. And a lot of it has to do too with like water temperature and things. But then I would sometimes find breeding in the big stones here, because this was more maintained, so they would clean it out and stuff, you know, all the trash and stuff. But there was still these boulders, so if it rained, it would kind of fill up here and then hopefully percolate in. But just like a lot of our Arizona flood control things, it would sometimes be enough time for mosquito breeding. So that's an example. Containers, a lot of those flower, you know, things on in cemeteries and stuff where they hold the flowers will sometimes have mosquitoes breeding in them too. So this was a gnarly site that took a long time. Here's some more potential sites. So this is a fountain, and just like green pools and stuff, anything that, you know, breaks down or is not maintained properly could become a potential breeding site. So this had a bunch of quinks breeding in it. And this was an interesting site. So this could be a potential site for like floodwater mosquitoes. But sometimes you'll see them, if there's water in the cracks, you'll sometimes find other mosquito breeding. But yeah, you could check this. And from the road or from the path, a lot of times these will look, oh, there's no standing water, but there'll be a little bit of water in the remaining of the cracks. This is another one of those ones that you may think is totally dry, but you have to walk close to it to see there's a little pipe that sticks up in a low spot. And there was quite a bit of breeding in here, and it was a mix of quinks and tarsals, which was kind of nifty. Another kind of flood control feature, it's hard to see, but there's an outlet here into a wash, and this has occasionally has a lot of water flow coming out of it, and actually dug out this, you know, eroded out this little thing. And this is that another view of that video where I was taking a dip, and there was tons of mosquitoes here. So you could have something that if it's not maintained, or if the conditions change, it may not have been breeding. A large storm comes up, makes a new site. And then this is just an example of a container I captured of some of the mosquitoes from up top here. And so that's, you're finally inside, you got some air conditioning on, and you're in the lab. So some of the things that you might be helpful. I can do basic ID in the field, maybe to genus, or at least to type, like floodwater, standing water, you know, anopheles are really easy. And so that's useful to know what products to apply. But in the lab, if you want to get to species, you're going to need probably some magnification. So like a stereo microscope. Nice thing, I use the same one that I use for my adult identifications. Some way of manipulating them, like a pipette dropper or something. This is one of those just like transfer pipettes. They're really cheap. I get like 100 of them so that I can kind of mess with them or cut them, or when I lose them. A little probe sometimes helps us, you know, to kind of nudge the larva. You want some identification keys, either developed in-house or books, white trays or little slides. I use a lot of times I'll use just to carry them from the microscope, either just a regular flat slide, or I like the ones that have like a little indent sometimes, so that the larva don't, if you tilt it, don't pour off the edge. You can also use like petri dishes, and those are all fine. Rearing chambers are really useful. I 3D printed this one because BioEquip's not around, so I was able to find a vendor with the jars, and then I just 3D CAD'd and printed my own rearing chamber. And that can be really helpful, especially as you're learning. What you can do is you can sort your mosquito larvae and then put them in different rearing chambers and then check to see if you're right. Especially if you're more comfortable with adult identification, you can use your sort of adult ID to confirm your larva ID. And then some way of recording, paper, electronic, or however. So here's another video. Just kind of how to manipulate. One difference with mosquito larva ID versus adult identification is they are alive and moving, so you're kind of trying to hit a moving target. What I'm kind of demonstrating here is the tip of this is too small to catch these fourth instars. So that can sometimes be good if you're trying to sort mosquito larvae into different rearing chambers so that you can especially if you're doing like insecticide resistance or something, you can pick off all of the pupa and they'll all emerge at the same time. And this is a big mix, so I have first through fourth instars and pupa here, so they can be hard to catch. What you can do is put them in the refrigerator a little bit, and that'll kind of cool the water down. You don't want to get too cold because they'll die, but you'll kind of anesthetize them a little bit, and they can be a little easier to manipulate. And then you can just transfer them to the slide. So I have both types of slides here. I have one of those ones with little indents. They're a little more expensive. The flat slide works perfectly fine. And then you'll just put your water here. If there's too much space and you're having a hard time looking at the mosquito under the microscope, you can immobilize them a little bit by drying off some of the water, which is what I'm trying to do here. And then I probably mess up and barf all the water out and have to, and then have to try again. But that is one way to move mosquito larvae. So some very basic, anatomy is useful if you're going through the keys. This is easy to find on the internet. I usually a lot of times print it out and have it stuck to the wall near my counting station. So you have your basic anatomy: head, thorax, abdomen, just like in adults. And then some of these head features are useful to know because they're mentioned. The antenna, the mouse bristles will sometimes look different. And then the thing to really key in on is this siphon. A lot of times the shape and the length will tell you the general groups that they're in, and then you'll have to look at like the combs, comb scales, and then the hairs will kind of help you get closer to species. Here's an example of kind of the three basic groups of mosquitoes. Saurophyra could kind of be grouped in with floodwater Aedes too, but you'll see they have a little bit different shaped siphons, and that's usually how to tell. Anopheles are really easy. You can spot them just in the water. They have no real visible siphon. They still have one. It's just very, very short. But since it's so short, they have to actually be almost parallel to the water line. So they're the way they just sit in the water is very Anopheles distinct. And then you'll sometimes see like Egypti or any Aedes has a little bit shorter, stubbier siphon, and then Culex will have this kind of longer, more kind of elegant siphon. And they also will hang at a little bit more of an angle, especially compared to Anopheles. So for like helpful guide and things, the University of Florida's Medical Entomology Lab has awesome pictures. And if you're kind of more of a line drawing person, that can be helpful. Or if you really like pictures that are annotated, the University of Florida has really good pictures. The only disadvantage of them is they don't have every mosquito that Arizona does. And we have some that they don't, they have some that we don't. So you may be missing a couple, but they have a genera key that can get you to genus, which could then help you use your books or your guides to get you to the species. And so the genera will be, those keys will be pretty much the same for both states. So I often refer to them. Awesome pictures for both adults and larvae. Highly recommend them for beginners and and more experienced. So here's a picture of some mosquitoes I was identifying. They are not all the same, so that's kind of the trick question. I believe these were Tarsalis, and then this one was maybe a Quinx, Orthothorax, I can't remember. But you'll be looking at, especially for these, comparing these two, these little tufts of hairs on the back edge of their siphon, and that can be one way to distinguish them. And just will definitely concede it is not easy. So here's what it would look like from like a key, and that's what I was referring to. So these little hair tufts here on the back, there's just slightly more of them, more groups of them. And then there are on the Quinx. So it can be very challenging. You will not probably be able to see that from the naked eye. Sometimes you can kind of see, especially, and it's worth mentioning, fourth instars is what is more easy to identify. It can be very challenging, if not impossible, with the lower instars, especially since some of these features don't develop until third and fourth instar. So, and then pupa, there are keys and guides for pupa. I often, admittedly, am kind of lazy, and if they're already pupa, I'll just wait a day and I'll let them emerge and I'll identify the adults. But you can use these same books for those too. But so yeah, it can be very challenging. You're going to be looking at these different, comb scale shapes, and hair patterns. And then different features, and so it's definitely very hard. It takes a lot of practice, and that's why I say, like, if you're going to be in the field or doing this, if you can get to genus, that is probably practical wise, as far as you need to go, just to know, hey, what product do I need. So you can use identification keys just like with adults. You can develop your own, or you can use some that already exist out there. This one's a pretty cool way of doing a key. And then what I'll often do is, like, you could just draw out here, go to your key sheet. So if you just label your key, like, this will take me most of the way, and then I want my distinct, especially if I'm using somebody else's key from a different area that doesn't have all of the species. So some of these dead ends, on this key, I may point to one of my other books or keys that I like to use. And then that can be helpful. And a lot of times I'll make my own little cheat sheets too. And then, so kind of the moral of all this, if we miss them in the water, this is what we get to deal with in the field. So it's definitely worth it. Definitely with the whole integrated pest management, mosquito management, we try to get them from all fronts. So if we can control them or eliminate the breeding sites, then it saves us a lot of work, in the field later on. And that's the end of that. So I guess we can take some questions.